coastal ecosystems, looking at mainly like the provision of and quantifying the provision of ecosystem services, primarily carbon dynamics in coastal and marine ecosystems, so mangrove, salt marsh, and seagrass. And he's done a bunch of work with Conservation International and other NGOs, in particular in Fiji and Indonesia, looking at carbon sequestration and greenhouse gas emissions in mangroves from both conservation and also restoration. So he's going to be joining us. He's going to be working on 3 projects here at the University of Queensland. One with Conservation International, looking at developing a restoration manual for blue carbon ecosystems, so mangroves, salt marsh, and seagrass, and another looking at quantifying the carbon stocks and seagrasses, and one more project which I can't quite remember, but I'm sure Clint will let you know. So we had a few tech issues, so we had to hand over the Zoom reins to Clint. So if you could please just make sure that you're all on mute, because Kate can't control anything, and then we'll have some time for questions at the end when you can unmute yourself and we can have a bit of a discussion with Clint. So yeah, you want to take it away, Clint?
Thanks, Valerie. Yep. So as with all these meetings, there's all sorts of technical issues. So I'm going to share my screen and hopefully it all works. Okay. So just checking. So can someone, Valerie, can you just check that that's coming up and you can hear my audio, please?
Yes, can see and hear.
All good. Right, you can hear me. Can you see me or not?
Oh no, we can't see you, but we can see your presentation. So
okay,
maybe we'll just have to see your presentation.
Yeah, you don't have to look at me. Yeah, so thanks very much for the introduction, and yeah, it's great to be joining UQ and working with Kath and the team here. And so this presentation, I thought I'd touch on some research we did in Myanmar at the end of 2019 and the start of 2020. So looking at rates of greenhouse gas flux from mangrove soils and what the implications are for forest carbon projects. And so this is a bit of context. So we know that mangrove ecosystems are both significant sources and sinks of greenhouse gases. So they can be either net sources or net sinks at different, across different time periods and different temporal scales. And because of that fact, it's crucial to really understand the variability and dynamics in flux rates. And that's so important in the context of land use change. In a country like Myanmar, which has one of the highest rates of mangrove deforestation in the world, it's up there with countries like Indonesia. The problem in Myanmar is that most of that mangrove conversion is to rice paddies, so instead of aquaculture ponds, which you see in the rest of Southeast Asia. So the initial aim was to head out to the mouth of the Ayeyarwady Delta there in the southeast, at a site that Kat's been working on for a number of years now, along with Sang, and would do this trenching experiment where you dig a series of trenches sort of 15 centimeters wide by 1 meter deep, and the idea is that you separate respiration from heterotrophs, from soil microbes, versus respiration from trees. And so the difference between the two can help you to get towards estimates of things like net ecosystem production. So that was the idea. And before I went out there, I'd said to our colleagues on site, can you dig some trenches for us, please, so I can do this study. And through some sort of miscommunication, when I got out to site, instead of digging trenches, they'd busted their backs and they dug these enormous holes in the ground. And so I got out there going, well, that is my entire experimental design gone. I can't do what I came out here to do. And these poor guys, they bust their guts, they dug these enormous holes in the ground, which I couldn't use. So yeah, so lesson one there was, the importance of making sure you're really, really clear in your communication. And so anyway, so I got out there and I realized that my entire experimental design was gone. So I thought, well, what can I do? And we ended up coming up with a kind of like a land use change design. So we've had these partially inundated aquaculture ponds. We've had a strip of established mangroves along the riparian margins, and we've also got these two planted forests of differing ages. And so I was running around there. This is day one of a 10-day field trip, and I figured out, well, let's do this land use change continuum and see what the difference in and greenhouse gas flux rates are between these different treatments, and from that we can kind of see how that changes over time. And we did the first assessment was at the end of the dry season in February, and then we came back out later in the wet season. And so the first pond, the dry manky ponds, this is I'll just hijack my presentation for a second. So the dry manky ponds are exactly that. They are dry, they are manky, they're smelly and they're dirty. And we actually ended up getting that name, dry manky ponds, into the publication that this presentation is based off. And that came through from a bet that my old PhD supervisor made to me. And he sort of said, Clint, if you can get the word manky published in an academic paper, I'll buy you a case of beers. And so I had to go through, Kath Lovelock and Lindsay Hartley, who are both eminent professors in their fields. And we managed to get the word manky inserted in this publication. So it was one of my finest achievements, but you can see why. Really dry, really cracked, and there's very kind of little growth there. The key thing to note, in the top right, in the top left corner of that photo there, you can kind of see some taller mangroves growing there. So have a look there, and also in the bottom left photo, where that blue tarpaulin is. So that's this, in the background of that is this 8-year-old rehabilitating site, and the foreground is this dry manky pond site. Now these two sites, this 8-year-old site and this dry manky pond, are separated by maybe a couple of meters. There's a low berm between them, and where I'm taking my chamber measurements, it's only maybe 2 or 3 meters apart. So the only difference, one's vegetated and one's got no vegetation on it. And you can kind of see the setup here. So so we're using the static chamber technique, which is basically I just use snap block container lunch boxes, and I cut the bottom out of that, and I insert that sort of 4 centimeters into the soil, and that's all airlocked and tight, and it feeds back into this greenhouse gas analyzer here. And so this is kind of day 2. I forgot to say earlier on. So when I figured out what I'm going to do. I'm going to do this land use change sort of design. But that night I got really, really sick, and as everyone who goes out to this project site does, it turns out, and I was sort of spewing and pooing all at the same time. All the things were coming out. It was just a massacre, to the point where Sang was on the verge of calling the Medivac helicopter. And so I had to go back out there and plug myself up the next day and get out there and try and get this field work done. And this is, so it's really tough work. It's 35 degree heat, and there's not too much shade shown by these mangroves here. But we got out there thanks to the help from our on-site collaborators, and managed to get through the next sort of 8 days. But it's tough after being that sick and that crook. Anyway, these are the results, and as you can see, there's not really much going on from any of the treatments. Our 5-year-old site, our established mangroves, our dry manky ponds, our partially inundated ponds, apart from this massive rates of carbon dioxide efflux from our 8-year-old site. So remember that this site is only separated from these dry manky ponds by 2 or 3 meters. So really, really high rates of carbon dioxide emissions. And if you look on the right-hand side of that graph, it's much, much higher than average rates of CO2 efflux from mangrove systems averaged across the world. So I scratched my head going, what is going on here? Why would that site be pumping out so much more carbon dioxide than all these other sites? We went back through the data. We went through it with a fine tooth comb, trying to figure out what's going on. In the end, we thought, we're going to have to go back out there and do a repeat measurement to get better data and see if we're seeing the same sort of rates occurring. And this time we went out with an actual decent machine that doesn't break down, using the same sort of chamber array, but just a different machine. So, but measuring the same sort of variable. So measures the rates of accumulation and parts per million and parts per billion for methane. So we went back out there at the end of the wet season, and went back to the exact same spots that I was previously. And this time, this is what we found. So every single site that I went to was going like the clappers. So rates of carbon dioxide emissions were just through the roof, and it was nuts. This is completely and utterly unexpected. Every single site. really high rates of CO2. Methane, although you can't see it on that graph, also increased between 50 and 450%. So really high rates of methane, but overall kind of negligible amounts. So this time I'm thinking, what have I done wrong? Have I mucked up the calculations, or is there a fault in the machine? And so again, went through the data with a fine-tooth comb, and yeah, it was a legitimate result. What the measurements we're seeing are actually legitimate. It's a real result. So you can see the comparison in this graph here between our February results and our October results. So all of these sites, there's just a exponential increase in almost an exponential increase in CO2 emissions between February, which is the dry season, and October, which is the end of the wet season. Even from our 8-year-old sites, which are already pumping out carbon dioxide during the February results, they increased. They didn't decrease. They increased in October. So it was completely unexpected, and it's definitely something that I wasn't too sure whether we'd done something wrong or not. And so we came back and thought, right, well, there must be an explanation for this. And so we'll start off with why soil CO2 emissions were so high from the 8-year-old site in February. And I think the most likely explanation for that is that it's related to primary production. So a growth spurt. So mangrove mangrove forests have been shown to go through these growth spurts between the ages of sort of 6 and 10. It's like a teenager going through a growth spurt, except they, instead of growing upwards, they kind of grow downwards, but they grow downwards by allocating a lot of biomass to below ground fine roots. And so those fine roots turn over rapidly in the soil as a form of nutrient recovery. And that forms a really large food source for soil microbes. And as they munch through all that organic matter, that CO2 is respired, and it leaches through these cracks to the surface. So that's what we think was happening during February, but why would that didn't explain what happened at the end of the wet season in October. So why were CO2 production so high then? And it wasn't until I went back and looked at the data, like rainfall records over the last sort of 10 years, that we learned that actually in the period between February 2019 and October, that should be 2020, when we came back out there was this epic monsoon event, so one of the largest on record. So a heap of fresh water came through, and it was actually caused a lot of flooding, and a few people died as well out there. So we didn't realize that at the time. And the thing with freshwater influx is that it lowers soil pore salinity, and you've got all this new alluvium which gets deposited. So the Ayeyarwady, it's got its source in the foothills of the Himalaya, and all that alluvium gets deposited. And freshwater influx sort of erodes that material and releases compounds which plants can take up. And because of that, it stimulates both autotrophic and heterotrophic activity. So plants respond to freshwater influx, and so do your soil microbes. And this freshwater influx there's a term for it called the Birch effect, which has been known about for several decades. And it's this short-term increase in respiration rates as soils cycle between dry and wet conditions. And the other thing, so with methane production, lower salinity, so freshwater comes in lower salinity, that tends to favor methane producing bacteria over methane consuming bacteria. So that's probably why we saw higher methane emissions. But as I said before, methane remained quite an insignificant source of overall greenhouse gas emissions. And so this diagram is just trying to just hammer home at that point. So basically, February dry season, got this growth spurt from the 8-year-old forest, and all of that's invested in below ground fine root turnover, which forms a large food source for heterotrophic microbes to consume and respire to the atmosphere. In October, it was extreme monsoon season, and that freshwater influx has been known to stimulate both autotrophic and heterotrophic respiration. And so we didn't measure things like bacterial populations, but just like a week ago, a publication did come out from the Sundarban in India and Bangladesh, like this one of the largest mangrove forests in the world, and they looked these two things. They looked at greenhouse gas flux rates, and they also looked at the populations of bacteria in the soil, and they found that sure enough, during the monsoon season, there was really high rates of greenhouse gas flux, particularly CO2. and that was related to really high populations of heterotrophic soil microorganisms. So, we're not just speculating, as these theories are, it turns out they are backed up by some of the science. So why is that important? Why would that be important? Why is it important to measure these really high rates of greenhouse gas flux? Well, what we measured out there, it was like the highest that had yet been recorded and published in the literature. So no one had measured rates of particularly CO2 emissions that high before, particularly using this static chamber methodology. The idea is that, sure, it was really high for that small limited period of time that we measured it, but surely in the long term that will be counterbalanced by sequestration and biomass in soils, provided mangroves remain intact. Those dry manky ponds, if they remain unvegetated, they're going to remain a net source to the atmosphere, net source of emissions. And it kind of, it hammers home what's so important in the context of land use change and when developing forest carbon projects to go out and measure these phenomena. And it's not just mangroves. Like there's a few publications that have come out that are looking at rates of greenhouse gas flux in other biomes, and particularly permafrost. So the birch effect, which we saw in our mangrove soils in Myanmar, so that's where the freshwater influx stimulates microbial growth, along with the rhizophora priming effect, where plant root growth stimulates an increase in microbial populations and this sort of symbiotic effect. Now that's also evident in melting permafrost. And that's even more worrying because you've got these previously frozen tundra soils, which are now unlocked, and they're unlocked so the soil's free for plant roots to grow. And that forms a source of liable compounds for the release of CO2 and CH4. And this rapid thawing is evident in these extreme climate change feedback loops. So you got melting permafrost driven by climate change. And because of that, that stimulates really high rates of CO2 emissions. So that's really important, and that's sort of juxtaposed against this other idea which is a recent publication from this year, where as temperature increases, our terrestrial forest carbon sink, the photosynthetic ability of our terrestrial carbon sink is decreased. So increased temperatures, plants reach a sort of a tipping point or a thermal maxima, where photosynthesis actually declines as temperatures increase. The problem, though, is that that's kind of outweighed by the fact that it's not seen in soil respiration. So at higher temperatures, the respiration rates from the soil from microbial activity actually increase, and that's what this graph in the top right is showing, which is a bit worrying. Basically, this publication is saying that under business as normal scenarios, it results in a near halving of the land sink capacity of forest by the year 2040 if we continue on the way we are. So that means at a forest carbon level, we can't really rely on forests anymore to soak up all the excess carbon dioxide, because their carbon dioxide sequestration ability is outweighed by the fact that microbes are spitting all that back into the atmosphere. And then if you think that through to a policy and a project level implication, there's a reason that I've got my undies on my face in that photo. I'll come back to that. So at a policy and project level, we've known about this for a while. So young boreal and temperate forests are often net carbon sources to the atmosphere. They're not net sinks, and that's driven by, like we saw in Myanmar, high heterotrophic soil microbial rates, and because they're in sort of colder climates, growth rates tend to be slower as well. They take a while to get up to speed and actually get to the point where biomass accumulation outstrips respiration from the soil. So this is not new. This is something that people have known about for a while. The problem, though, is that doesn't translate to at the policy and the project level. And I know this because I looked through during my PhD research. I looked up would have been about 100 forest carbon projects registered on the verified carbon standard, and I looked at their emissions reductions, what their project was claiming in terms of carbon sequestration, and all of them looked at basically biomass accumulation. So forest carbon projects, biomass accumulation, change in biomass over time. Some of them looked at soil carbon burial, but none of them considered the fact that actually these forests also emit also emits carbon dioxide to the atmosphere, from soils to the atmosphere. So none of that was accounted for. And that's really important, because when you plant a tree, it doesn't exist in isolation. It's part of the ecosystem you planted in, and that includes the soil. So if you're not accounting for respiration from the soil, does that mean that we're overestimating our net mitigation outcomes? And that, if you think it through to, so forest carbon projects, they last, their typical span is about 30 years. And if you're doing them in boreal and temperate forests, even mangrove forests, which is what we saw in Myanmar, does that mean that actually for the first sort of 10 years in certain biomes, what you're claiming is your carbon credit removals and reductions it's actually a net source. It's not a net sink. That's fundamentally important, and that's why it's so important to get out there and do these studies using these static chamber techniques, or even better, using methods like eddy covariance towers, where you can just install a tower and that measures the changes in carbon dioxide between over annual periods. Really, really important. So that's the importance of this. This is what's so important, to get out there and do this sort of work. And I'll finish off with so I'm wearing undies on my head here, and I thought I started off this presentation with a tale of woe from the fieldwork out there, and it's probably appropriate to bookend it with a tale of woe. So as I said at the start, I got really sick out there on the first day, and and my entire experimental design was, had to think through an entirely different scope. Anyway, so we got through the field work, and I thought, right, we've done 8 days of, I'm going to go back to the hotel and I'm going to have a beer. I'm going to relax. 2 days just to unwind. And somehow, while I'm having my cold beer, celebrating the end of field work, I've got a scratch in between my eyelid and my contact lens, and so my eyes just weeping out with tears, and I can't see anything. I have to spend the next 2 days inside because I can't go outside because the lights are so bright. It was so bad. So that was the joys of fieldwork out there. Lovely people, amazing place, amazing experience. But one of those bits of fieldwork where you're just so happy to see the end of. Okay, with that, I'll pass over to questions and comments.